tell us about your work and the conclusions, and then we'll go to the questions. So, as you
The title of the thesis is A Complex Network Approach to Phylogenetic Trees from Genes to the Tree of Life. And this work has been supervised by Professor Emilio Hernández-García, Dr. Víctor Eguíluz, and Professor Duarte. This work stems from two different motivations. On the one hand, our interest in the comprehension of evolutionary processes, and on the other hand, the huge expansion of that complex network theory has undergone during the last decade. Both inspired us to apply complex network theory to the inference of evolutionary patterns. In that sense, the two main purposes of this work has been, one, to provide new tools within the context of complex network theory for the topological characterization of evolutionary trees, and the second purpose was to contribute to enlarging the knowledge of evolutionary patterns depicted in them. With that aim, we use as a theoretical foundation from complex network theory the allometric scaling approach. Allometric scaling characterizes how biological quantity scales with the size of a biological system, such as, for example, metabolic rate or lifespan as function of the body size. This approach during the last decade has also been applied and extended to the characterization of the relationships between the shape and size in tree-like networks, such as river basins or also blood vessels or bronchial trees. Taking this theoretical background in account, we decided to extend this approach of the allometric scaling analysis in tree-like networks to the characterization of another tree. This is the tree, the evolutionary trees, the tree-like representation of evolution, such as here got an example of a tree of life. And for this, we proposed a measure for the characterization of the tree size, and we call it branch size, or A. And we proposed also two different measures for the characterization of the tree shape. One is the cumulative branch size, and we will call it C, and the second is the mean depth, or D. The branch size, we define the branch size as the number of subtaxa that diversify from a certain node in a subtree or tree. So, for example, for this tree, the branch size of the tips is 1, while for the internal nodes, the branch size, or A, is for this subtree of 3 nodes, A is equal to 3, for this is 5, and for this is 7. The cumulative branch size, on the other hand, is defined as the sum of all the A values for the different nodes that build up a certain subtree or tree of life, or phylogenetic tree. In that sense, all the tips, they have a C value 1, while for this subtree of 3 nodes, since the A values for the different nodes is the two of the tips, they have A value 1, and the internal node has A value 3, the C value for the internal node is 5. For this internal node, the C value is 11, and for this is 19. The mean depth, we define the mean depth as the average depth over all the nodes that constitute the phylogenetic tree. Defining the depth of a node as its distance to the root, such as, for example, for this node, this one is 3, and the depth is the distance to the root plus 1. So, while for this node the distance is 3, the depth is 4. The relation between the two measures for the characterization of the tree shape, the cumulative branch size and the mean depth, is given by this identity. And the mean depth corresponds to C over A. In order to show which is the ability of this measure for the characterization of the topology of the phylogenetic trees. I plot here 3 different trees with different topology, but with the same size. So, on the one hand, we have a fully balanced tree. In example B, we have a fully unbalanced tree, and C corresponds to a real phylogenetic tree. The fully balanced tree shows that it depicts the lowest values for C, while the largest values for C was observed for the fully unbalanced. And if we focused on the scalings for these measures of the shape for the 2 extreme topologies, what we observe is that for a fully balanced tree, the C and D scales logarithmically as function of A, while the fully unbalanced tree, what we observe is that C follow a square A dependence, while for the D shows a linear dependence with the branch size. Taking this theoretical background, what we decided was to apply the depth scaling analysis to different kinds of evolutionary trees. So, the first section of this talk, I present the results detailed in chapter 3 of the thesis, and they correspond to the depth scaling analysis of organism phylogenetic trees. In section 2, we extend this analysis of the depth scaling to the characterization of gene phylogenies that are presented in chapter 4 in the thesis. In section 3, what we decided was to try to propose a model what could help us in the comprehension of which are the biological mechanisms that reproduce the patterns observed in organisms and gene phylogenetic trees. And this was presented in chapter 3 of the thesis. In section 4, I extend this depth scaling analysis to the characterization of taxonomic trees. It was presented in chapter 6 of the thesis. And finally, in section 5, I will propose a depth scaling, a branch length scaling of phylograms, and it was included in chapter 7 in the thesis. So for the first section, our main goal in the analysis of the depth scaling of organism phylogenes was to try to characterize how preserved are the branching patterns across the different evolutionary scales, macro and micro evolution. And for that aim, we compiled 2 different kinds of data sets. On the one hand, interspecific phylogenetic tree data sets, and on the other hand, intraspecific phylogenetic trees data sets. And for this, for the interspecific, what we used was the tree-based data set. That this is a huge public compilation of different phylogenetic trees, and we downloaded more than 5,000 of phylogenies. And for the intraspecific phylogenies, since there isn't any public database of were compiled intraspecific phylogenetic trees, what we used is was the manually compilation of 67 phylogenetic trees, intraspecific, and in order to see if it were comparable, this large set of phylogenetic trees with this small set of phylogenetic trees with this large set of phylogenetic trees, what we did was to compile manually a set of interspecific phylogenetic trees, and these manually compilations for the inter and intraspecific were done with the idea of integrate organisms from the main different environments, climatic regions, and branches of life. And those trees were reconstructed with 3 of the main phylogenetic tree estimation methods: neighbor joining, maximum parsimony, and maximum likelihood. The first analysis that we did was the characterization of the branch size and the cumulative branch size distribution. And for that, we analyzed the cumulative complementary distribution function. In both cases, for the branch size and for the cumulative branch size, both functions displayed a power law scaling, and both data sets, the intra and the interspecific, show similar scaling for the branch size distribution and for the cumulative branch size distribution. The similarity between exponents between intraspecific and interspecific was also observed in the allometric scaling. And as an example, here I show an example of a phylogenetic tree of intraspecific, and this corresponds to different strains of bacteria Vibrio magnificus, and this is its corresponding allometric scaling. And on the other hand, as an interspecific phylogeny, I show a phylogeny of species from the order Cirudiformes and its corresponding depth scaling, and we see how similar are the different allometric scaling. And this resemblance between the different exponents is highlighted if we compare the probability distribution of the power law exponent theta, in both cases that was fitted for the individual trees. And what we observe is that a very narrow range in the probability distribution for both cases. And plotting together now the two different interspecific data sets, the one manually compiled and the one from trees, what we observe is that for the branch size, for the cumulative branch size distributions, and also for the allometric scaling, both data sets matched in the scaling exponents for similar behavior. In order to characterize or to compare which is the behavior of the real data with the behavior of a random tree, we used a classical model that is the equal range Markov model, and what we observe is that the real data deviate from the random behavior, especially for large sizes, while the random phylogenetic tree, the random trees showed a logarithmic dependence with the branch size, which implied a different scaling from the scaling displayed by our empirical data of intra and interspecific phylogenetic trees. From these results, we conclude two main ideas. The first one is that the similar allometric exponents suggest the conservation of branching rules across the entire history of life. And the second conclusion is that the deviation from the random model suggests the operation of mechanisms generating correlated branching. With the aim of extending this approach to the analysis of gene phylogenies, in order to characterize how preserved are these branching patterns across gene and species evolutionary levels, what we did was the compilation of a gene phylogenes dataset, and for this we used the public database Pandit. We downloaded close to 8,000 phylogenetic trees, and we compared this dataset with the one used in the previous section for the interspecific organism phylogenetic trees from Treebase. And the analysis of the depth scaling for the organism phylogenetic trees, what we observe is that this scaling suggests a square logarithmic scaling with the branch size, and was lying between the two extreme topologies. On the one hand, the linear dependence of the fully unbalanced phylogenetic trees, and on the other hand, the linear dependence, the logarithmic dependence of the fully balanced trees. In order to check how preserved is this average behavior analyzed for the whole Pandit database, if we discriminate between the different plotting functions, what we did was to select a set of phylogenetic trees with different functions, and what we observed is that the depth scaling of these protein phylogenies with different functions shows the same scaling behavior as the one depicted by the whole Pandit database. And this generality in the exponent was confirmed or reinforced after comparing the depth scaling of protein phylogenetic trees with the one described before for organism phylogenetic trees. And what we observe is that both data sets display very similar depth scalings. And taking into account the result of the previous section, where we described that organism phylogenesis was fitted to a power law, that's such as this graphic, but here we proposed a square logarithmic fitting for the protein phylogenesis. What we observe is that the square logarithmic scaling is offered a similar fit for organism phylogenetic trees, while a power law scaling is not so good for protein families, and is better a square logarithmic scaling for protein families. In the first analysis of the protein phylogenesis that I showed, we observed that in the raw data there were some dispersions from the average behavior, but these dispersions are restricted only to certain regions of the phylogenetic tree. So, for example, this is an example of a tree with these dispersions. And what we observe is that the average behavior of the tree corresponds to the average behavior of the whole database, while only the dispersion are given to certain regions where they depict a presence of unbalanced trees, a high presence of unbalanced branchings, and this highly presence of unbalanced branchings corresponds to this region of this tree. From this result, the two main conclusions that we obtained was that the branching and scaling patterns in protein families do not differ significantly from the patterns observed in species phylogenesis, and the universal scaling properties of gene and species level suggest the universality of branching rules across the entire history of life, from genes to species. Now, with the aim of finding a biological explanation to this universal scaling that we obtained for the organism and protein phylogenetic trees, we proposed here a model which depicts a depth scaling similar to the one depicted by organism and gene phylogenesis. The use of models for the characterization of the branching of phylogenetic trees, it has a long history, and the first model proposed was by Ure in 1924, and this was the classical random model, and it is based in the birth and death processes, which consider a birth rate and a death rate, and the special case that considers Ure for the characterization of the distribution of species inside genera was consider this process as with birth rate constant and the death rate equal to 0. And for this distribution, what he obtained was that the distribution of the number of species inside genera followed a geometric distribution. This model was continuous in time, but some decades later was proposed a random model, discrete in time, and it was called equal rates Markov model. This is the one that we used in the first section. And in this model, starting from a single ancestral species, one leaf is chosen at random, and among the different tree leaves existing of the present time with speciate. But as we described in the first section, random model depicted a logarithmic scaling with the branch size, while our data doesn't correspond with the same behavior. And that leads to the proposal of different models and different scaling for this branching process. And for example, Aldous proposed the AB model, which shows that the phylogenetic trees generated with this model displayed a square logarithmic dependence of the mean depth with the branch size, while Ford later, some years later, proposed the alpha model, which shows a power law dependence of the mean depth with the branch size. And some years ago, Stephan and Rubia proposed an alternative explanation, and they claim that the non-random scaling of the real phylogenetic trees were related with small size transient effect, where for large sizes of the phylogenetic trees we will observe the logarithmic scaling again. The model that we propose is the evolvability model, and this is based in two evolutionary possible outcomes. On the one hand, evolvability, on the second hand, robustness. So, for a given species, all the species that are able to speciate at a certain time, all them they will speciate, and each species will give rise to two new species, and this new pair of species will suffer two possible outcomes. On the one hand, with probability p, the new species have the same probability of model species to speciate, and on the other hand, with probability 1 minus p, one of the daughter species is unable to speciate again. This model has the property of combining symmetric branching and asymmetric branching processes, what imply the introduction of some correlation in the branching process, since for the asymmetric branching, one of the species is precluded to speciate again. But this correlation disappears for large sizes, observing this the random scaling for large sizes. So confirming obtaining again also the transient behavior described by Stephan and Rubia. So if we analyze the trees generated for this model, what we obtained was that those trees with generated with p equal to 0 will reproduce the scaling of the fully unbalanced trees, while those trees generated with p equal to 1 reproduce the scaling of the fully balanced trees. And the real data of protein and organism phylogenes were reproduced with the trees generated with p equal to 0.25. From this section, what we conclude is that the evolvability model shows a non-random behavior as a transient for small tree sizes, with the logarithmic behavior of the random scaling as asymptotic behavior at long sizes. The evolvability robustness interplay highlights the effect over the depth scaling of the presence of asymmetric diversification events. And finally, the incapability to diversify again can be found at macroevolutionary level, such as, for example, taxa that require very long refractory period, or random massive extinction of taxa. And on the other hand, at microevolutionary or at gene level, we're going to observe also this incapability to diversify again in individuals of a population, or genetic variants from a cell, embryo, or individual that are unable to diversify. In the fourth section, our main goal was to characterize which was the effect of the different taxonomic classification criteria over the branching properties of evolutionary trees. And from this, we focused on the two main taxonomic criteria. The first one was the rank-based taxonomy, and it is based in predefined taxonomic ranks. The 8 major taxonomic ranks are the domain, kingdom, phylum, class, order, family, genus, and species. And on the other hand, as a rank-free taxonomic criteria, this taxonomic criteria is based exclusively on evolutionary criteria. So, for example, if we focus on the taxonomic tree of fungi and its corresponding phyla, if we are based on rank-based taxonomic criteria, what we obtain is a fully polytomic tree where the different phyla are directly connected to the root. And if we represent this tree with a rank-free criteria, what we obtain is that we observe internal binary bifurcations that correspond to specific evolutionary relationship between the different phyla. For this analysis, we use the taxonomic tree of catalog of life, which includes more than 1 million of species from the different kingdoms, Animalia, Archaea, Bacteria, Chromista, Fungi, Plantae, Protozoa, and viruses. And the taxonomic ranks that it considered were kingdom, phylum, class, order, family, genus, and species. As a rank-free taxonomy, we use the taxonomic tree that is built by the Tree of Life web project, and it includes more than 25,000 of organisms. And we compare this in order to compare the taxonomic scaling with the phylogenetic trees. We use the data set that we used in the previous sections for the organism phylogenies that were obtained from Freebase. The first analysis that we did was the characterization of the branch size and the cumulative branch size. And for this, as we did in the first section, we used the cumulative complementary distribution function, and what we obtained was a power law distribution in both cases, for the branch size and for the cumulative branch size. But the different data sets, the rank-based and the rank-free taxonomic trees, they show different exponents for both measures. And putting together the exponents of the distribution for branch size and for the cumulative branch size for the phylogenetic trees, and the one depicted for the rank-free taxonomic tree and for the rank-based taxonomic tree, what we obtained was that the rank-free taxonomic tree showed a similar exponent to the one depicted by Freebase. The analysis of the depth scaling of the taxonomic trees showed again a discrepancy between the different taxonomic criteria. So the Tree of Life, the rank-based taxonomic tree from the Tree of Life, the web project, displayed a depth scaling closer to the one depicted by the phylogenetic trees from Freebase, while the rank-based taxonomic tree from Catalog of Life displayed a mean depth scaling as function of the branch size closer to the scaling of a fully polytomic tree. And this similarity with the behavior of a fully polytomic tree can explain, taking into account that the rank-based taxonomic criteria consider a fixed depth in the classification. So regardless the number of organisms that you are considering, the mean depth is approached constant. So while for a fully polytomic tree the mean depth is 2, for this rank-based taxonomic tree of Catalog of Life, the mean depth is 8. In order to characterize if this average mean depth scaling was conserved in the different kingdoms of organisms, we discriminate between the different kingdoms of organisms, the depth scaling analysis, and what we obtained was that those kingdoms with larger presentation, such as Animalia, Plantae, and Fungi, they depicted a mean depth scaling similar to the one depicted by the whole taxonomic tree, while those kingdoms with lower representation, such as Chromista, bacteria, protozoa, and viruses and archaea, show some deviation from the average behavior, which is related. They depicted a mean depth scaling closer to the one depicted by the rank-free taxonomic tree or the phylogenetic trees. In the second part of this section, taking into account that the number of connections that a node has is measured by the degree of this node, what we analyzed was the degree distribution and how is this degree distribution distributed all over the taxonomic trees in order to analyze the prevalence of polytomies in the taxonomic trees and how are these polytomies distributed all over the taxonomic trees. And the analysis of on the cumulative complementary degree distribution displayed that the taxonomic trees, for both taxonomic trees, displayed a wider degree distribution than the one depicted by the phylogenetic trees from freebase. So while for the phylogenetic trees from treebase, we were not able to find nodes with degree larger than 66. For the taxonomic trees, we observed nodes with a degree very large. So, for example, for the rank-based taxonomic tree, we could find nodes larger than 8,000 degree, while for the rank-free criterion, we observe nodes larger than 400 degree. And if we focus in the probability distribution of the degree for these trees, we observe that the rank-free taxonomic criteria and the phylogenetic trees show a high presence of nodes with degree 3, which reflect their tendency toward a binary topology. In order, after having analyzed the degree distribution, we wanted to analyze the correlation of the degree with the depth of the evolutionary trees. And what we obtained was that while for freebase and rank-free taxonomic criterion, we didn't find any dependence on the degree with the mean depth. What we observed was that for the rank-based taxonomic criterion, we observed some dependence with the depth. And given that for the catalog of life, for the rank-based, this is the construction is based on rank-based taxonomic criterion, that we were able to identify each taxonomic rank with a certain distance to the root. We characterize the average degree of each of the different ranks of this taxonomic tree. So plotting the average degree with the different as function of the different ranks. So for example, at distance 1 we found the rank of kingdoms, at distance 2 the rank of family, and so on. At 4 as second is the distance 2 is the rank of phylum, at 3 is class, 4 order, and 5 family, at distance 6 is genus, and at 7 is the rank of species. What we observed was that the taxonomic ranks with higher presence of degree was of a high degree, and polytomies was the ranks of kingdom, family, and genus. And now, with the aim of characterizing in which manner the different kingdoms of organisms contribute to this polytomic nature of the taxonomic tree, we discriminate between the different kingdoms of organisms. And what we observe was that those kingdoms with larger representation, again, Animalia, Plantae, and Fungi, were the kingdoms that contribute more for the polytonic nature observed in family and genus ranks. While those kingdoms that contribute to the highly polytonic nature of kingdom rank were those that were the protozoa, viruses, Animalia, Plantae, and bacteria. And a third remarkable result from this analysis was the low contribution to the polytonic nature of this rank-based taxonomic tree from archaeas. And this can be related to the fact that this kingdom has a very low representation in this database. So from these results, the two main conclusions that we obtained was that the rank-based criteria distort the branching pattern of evolutionary trees considerably, while rank-free criteria are more respectful with it. And second, the perturbation effect is especially reflected in the taxonomic ranks of genus, family, and kingdom, as well as in the kingdom of organisms with larger representation. Putting together the results that we obtained in the first section and in second section about this universality in the depth scaling analysis of protein and gene phylogenies, with the analysis of the depth scaling that we did here with the characterization of the taxonomic trees, we can conclude that the universality of the results reported in the analysis of organism and gene phylogenies is not a consequence of the analysis methodology used, but it is a property of the biological datasets. In our last section, we tried to characterize the branch length distribution all over the tree of life. And for this, we detailized the tree of life of the time tree database, which is a starting project which includes only the leaves of this tree, corresponds to more than 1,600 families of organisms of the different three domains or superkingdoms, Eubacteria, Archaea, and Eukarya. And the first characterization that we did was the branch length frequency distribution, and for this we analyzed the cumulative complementary distribution function of the branch length in millions of years. And what we observed was that this distribution displayed two exponential decay regimes with an inflection point around 200 and 300 millions of years of length. Analyzing the branch length frequency distribution for the different groups of organisms, what we observed is that the exponential distribution for the different groups of organisms displayed a single characteristic time for each of those, what make us to conclude that this double exponential behavior was given by the combination of the different exponential regimes from the different groups. In order to characterize how the branch length is distributed all over the depth of the time of the time tree, what we plot here was the length of each branch that we found at each time period. And what we observed was a positive correlation of the branch length with the age, which was contrasting with the random model, where for a random model there is no correlation with the branch length. It's remarkable, but this positive correlation was highlighted if we erase the branches that connect the leaf, and this was given by the fact that while most of the branches are very short in the tree. That those branches, this is a representation of the branches that connect the leaves to the tree. Most of the branches are connected to that connect the leaves are very short. There are numerous branches that are very long, and this is because they split very soon, as we can see here in the tree, and that makes that at each transect of time we will find this length, and that makes that the correlation is turned down if we included these branches. So from these results, the conclusion that we obtained was that the frequency distribution of the branch length in the tree of life displays a double exponential decay derived from the two different exponential decay rates of the different groups of organisms in the tree of life, and the branch length distribution shows a positive correlation with each. But we recognize that we need improved version of the time tree database for extracting ground biological conclusions. The results that I presented here were I focused, due to time constraints, I focused on the results presented in the main chapters of the thesis. But I included also in the thesis some appendix where I extrapolate different ways or extended results from the depth scaling analysis. So in Appendix B, I included the analysis of the effect of the outgroup or the branching patterns of organism phylogenetic trees. In Appendix C, I present we try to characterize which is the contribution of speciation and gene duplication in the diversification of protein families. In Appendix C, we included a model which is characterized by depicting a non-random depth scaling behavior. In Appendix E, we included an extension of the evolvability model with including refractory period and mass extinction events. In Appendix F, we extrapolate the comparison between rank-based and rank-free taxonomic criteria to the characterization of language taxonomies. And in Appendix G, we propose a set of measures for the characterization of the depth scaling, taking into account the branch length of that are given by the information of phylograms. So, as summarizing, we resume here in 4 conclusions our work. The first conclusion is that the similar scaling patterns in intraspecific, interspecific, and protein phylogenes suggest the conservation of branching rules across the entire history of life, from genes to species. The evolvability model reproduced the scaling properties displayed by the organism and protein families, showing a prevalence of asymmetric diversification events. The rank-based taxonomic criteria showed a very distorting effect over the branching pattern of evolutionary trees, while the rank-free criteria proved to be more respectful with it. And finally, the time frame of life showed the exponential frequency distribution of the branch length, as well as a positive correlation of branch length with age. That's all right.
Okay. Thank you very much for your talk. And now it's time for questions. And I would ask Dr. Joan Pons, please, to start with 15 minutes.
Well, first of all, thanks for inviting me to be here. And I'd like to congratulate the candidate and directors of this job. I think they did a great effort to try to join 2 very different areas of science, like biology and science. And that can be a difficult work to do, because sometimes you are not going to, not going to be happy the biologist, neither scientist or physicist. Well, I told you many times all these topics before and different talks. And first of all, I would like to to talk about the database, the tree base you use.
Yeah.
I mean, my main concern about using this kind of meta-analysis is that you don't know the quality of each individual cases. And most of all, we never know the true history. The true tree is always unknown.
Yeah.
That is the big problem we have.
Okay.
And the better approach we can do is make the better hypothesis we can do with the data we have.
Exactly.
And we have limited of the amount of data, the sampling. I mean, what do you think about
Well,
taking this or trying to find universal scale based on these specific histories, and each one has to have different problems, shortcomings?
Yeah, but I'm with you that there is a problem that we are not able to know the truth, but in everything in life we are not able to trust ourselves. And starting from this point, what we did was, well, let's see what the biologists of the field biologists publish, and then we use the results that they publish in recognized journals. And taking, I know that that's a problem, but this problem is also for the biologists.
I tell you that we disagree
No.
about ourselves, about the hypothesis, and we are saying you are wrong, you are right. I mean, it's not the same, the quality of the trees that were published 10 years ago, than the trees that are published nowadays, and you have a better models of evolutions, we have a better reconstruction models. You put everything on the same.
No, but everything that...
Well, everything that has been published, I there. You don't know if that quality is good or bad.
This work started with my manually compilation of 300 of trees, and I counted by hand all of them, and these were compiled taking into account the recent publications from the last year. And we analyzed the scalings, and we observed some universality. And then we said, let's see what happens for larger sets if we were able to find them. Then we found tree base, and we analyzed the tree base, and we obtained the same behavior. I'm with you that maybe there are some problems with the data, and that's all. But the question is, there is some universality from the global point of view.
Well, that is my main concern. You can find any universal law if you start with data that you are not sure that is correct.
But then you are saying that there is no result from the biologists that you can trust.
No, we don't have the true trees. Well, we have the better hypothesis for sure.
The better, and you do what you can do.
Well, the problem is we have the true trees. Our job is done. I have to find another job.
And then you have to trust it. You say, I don't have the real trust, but I have the best result that I can have. But that's not the question. The question is the point is that we obtain some universal scaling using different reconstruction methods for the different organisms. And then the question is, this universality is the result of what? There is a biological reason, there is a methodological reason. I don't know, because maybe the different reconstruction methods include they have the same bias for the reconstruction analysis. I don't know. But I would think that at least now in the tables we have the two different possibilities.
Then another concern I have is like you differentiate very clearly what is a species tree, what is a gene tree, interspecific. So, well, in a sense, all are the same. They are gene trees. And we assume that gene trees represent the true history of that species. That's true, no? I mean, we don't have a species tree, because that would be the true tree. We have to know back and see how they evolved on that time. What then we can do is use molecular sequences or morphological characteristics
Yeah.
to be matrices,
Yeah.
and they use this method of phylogenetic reconstructions, trace back, and try to see what happens.
Yeah.
That's an inference.
Yeah.
I mean, but all of them, they are gene trees or characteristic trees or whatever
Yeah.
you want.
Yeah.
But they are not these species trees. You cannot differentiate them. For that reason, you find the same pattern in each of all of them, because all of them, they are species. They are the same, gene trees.
Yeah.
Or not.
No, but let's. No. I'm with you.
Right? I mean, they are gene trees.
Yeah, for sure. They are. That's basic. But within the, if you focus on the gene trees, you can distinguish. Let's say that an organism tree is a fully orthodox tree. Gene trees.
All of them are orthogonal, that's for sure. All the trees are based on orthogonal, on the same gene, otherwise that is a mess.
No, of a species, yes.
Yeah.
But of protein, no.
No.
In protein phylogenetic, you are including parallel
Yeah.
bifurcations.
You're right.
So
You're right.
then this.
But this is the same pattern. It's like a speciation.
No, you don't
duplication is like a speciation path.
But you
Point
don't
of
know
view.
that.
I mean, it's the same property.
Well, you don't know that.
Well, it's
You have to
duplication,
compare,
like
no.
a split.
No,
it's
but
for
for
that
that,
gene.
no, but for that you have to compare a fully orthodox tree
Mhm.
with a fully parallel tree
Yeah. you
and
can
see
compare
if the
it.
branching pattern is the same or not.
Yeah, it's going to be the
But
same.
if you, yeah, but this is
Or not.
no, I think so. But you cannot say no. This is
But
this is the same.
you prove it that it is.
Yeah, but I couldn't find any fully parallel tree or a fully orthodox tree. But I'm with you that an organism phylogenetic tree, yeah, is a gene phylogenetic tree, but the evolutionary processes that you represent in the phylogenetic tree, they are different.
Yeah, but you inferred from the same data.
Yeah,
It's
and
not,
what?
because it's not the species tree. The species tree doesn't exist. I mean, it's gene trees. All of them.
If you analyze whole genome trees,
Well, there's no that type of. Well, maybe in the future it will be that kind of analysis, but not now.
well, there are some trees of whole genome trees.
Full
When
genome
we
trees.
analyze them, but there are only a couple of trees because there are thousands of data or words only representing a small tree of whole genomes, but there are a couple of that they are large trees. I don't know. There is a published paper in Science of the tree of life of the whole genome organisms that are, well, the basic ones. The Drosophila archaea, the different bacterial archaeas and some plants. But I don't see your point. What do you mean? With the organism phylogenies are gene phylogenies, but
Yeah. I mean, no, they are all the same. All the same are based on genes.
yeah, but you cannot compare. Yeah, but you cannot compare the. Let's say you have a set of proteins that they are in a certain organism, but you cannot compare this set of proteins with the set of species of this organism. Because if this set of proteins are fully orthodox, you can compare them.
Yeah.
But if not, you are including.
Well, you cannot use them to infer a species tree, but it's an inference. You
Yeah.
make it.
Yeah.
Gene tree, and then
Yeah.
from that gene tree you can infer whatever you want.
Yeah.
And we infer evolution of a species.
Mhm.
Or you can make a gene tree and then infer the evolution of the genes.
Exactly.
But they are all trees based and built on genes. That means
Yeah, but you have said
for that they have the same pattern. Are you sure? I mean, or they have the same scale.
No. What we want you to analyze is different evolutionary processes, as you said. On the one hand, organisms, and on the other hand, proteins.
Yeah, the scale is the same, but
Yeah, and the scaling, you mean that this scaling is due to the fact that those are gene data?
well, in fact, well,
In
that
fact-
on the reason is because they are
Yeah.
related, that's for sure.
Yeah. There is Morphogan, it's a database of, you know this database of morphological tree
Mhm.
reconstruction? Well, there is problem with the number of trees that there are inside, but I downloaded the one that I put, for 15 trees from, basically most of them they were focused on facts, phylogeny. But we analyzed them and they show the same state in behavior. So I am with you that we have problem with the data and everything, but if you are, if you don't trust in our results, the problem is not from our point of view, the problem is of the data that we have.
Okay.
And this is a problem in biology and in our work.
Well, if you could go to the pandit, the slide.
Yeah.
The one with, this one. This one. The most interesting the main concern of all this is, for me, for my understanding, that the main scaling you find is driven because you are working with binary trees. As we discussed it many times and many times.
Yeah.
Here you can see a very interesting slide that the fully resolved,
The fully balanced?
The fully balanced and the one that is
Fully unbalanced, yeah.
Fully unbalanced. Okay? And you can see below the your pattern, there is very few. No? And
sorry.
And here,
those ones.
Yeah. See this part of the tree?
Yeah.
The graph. This part of the graph.
Yeah.
There is where you should find most of the trees that have polytons.
But in fact, those that are this, because those are binary. All of them are binary. But maybe you can find some of them that are fully binary. No, not fully. But
you can see that
you can find polytomies.
No, but one thing I can say that is very narrow.
Yeah.
The part of the graph that you can with binary trees is very narrow.
Yeah. With binary trees is very narrow.
Sorry. No. But with the scaling is very narrow because all of them they are only on this part of the graph. You cannot find part of these trees outside of these parts. They are the boundaries, no?
Yeah.
And even more, below, you can find very little because most of them should be polytomies. All the time you are working on that part of the graph, the up part of the graph, because you force it to be. But to be, in your data set, most of them are binary trees.
Yeah.
Well, is 0.52 and the perfect balance is 0.5.
Yeah. But which is the percentage of nodes that are polytonic in a phylogenetic tree?
Very few.
Then
you forced it to do so. You force it to do so.
But we are again in the same question. Then you cannot trust the data in biology.
No. That is the way we represent that.
Yeah, but after getting these results, you infer some conclusions.
That's for sure.
So you have to trust this data.
Yes. But this power scaling, the main thing is, when you use the call, you don't find this scale law. No. The rank.
Yeah.
You don't find it.
Exactly.
What means that? All the time you use it and check the pattern, you find this power law. Except in one. And the one is the characteristic of this one is having full of polygons.
Yeah. But no.
For my understanding, that could be an artifact of the.
No, for sure. But usually the fully resolved the polytomies, you can distinguish between hard polytomies and soft polytomies. And usually the soft polytomies are related with artifacts in the reconstruction method. So you are starting from the hypothesis that a fully resolved phylogenetic tree is a binary tree in biology.
Well, you try to, otherwise.
You try to get a fully resolved tree, considering that a fully resolved tree is a binary tree, unless you have hard polytomies that are related with radiation and everything. But most of the nodes are binary in biology.
Yeah.
So if you consider that most of the nodes are binary in the phylogenetic trees, it's logic that most of the phylogenetic trees that we observe are
But
binary.
I agree with you, but that means
Another point, sorry. The polytomies in the phylogenetic trees represent only one node. That implies that in this representation is nothing. It's an epsilon from all the data sets. So the average is the same, but if you have a polytomy, maybe this node is below the scaling, but it's only this node, not the whole.
No, I agree with you. I mean, the pattern you see is because we force our phylogenies to be binary. I'm not saying that is not true. We force it to do it, to be so. And then it's going to be binary. For example, the scaling you find is not a biological property.
Yeah.
It's because we force the trees to be binary.
Yeah, but this is cyclic. Then
No, it's not cyclic. But we force it. I mean, it's like.
But if you force, that means that you cannot trust the results.
No, we trust the results, but it's the way we represent.
Yeah, but why do you represent in this way?
Well, because from a philosophical point of view, from cladistics, you assume that only possibility of diversification is a binary split. But
Why?
well, yeah, because it is the most simple explanation. It's the most simple. I mean, have you found that in many places you have like a large split population, and they radiate at the same time, radiations? It can happen. It can happen. But most of the times that's right, it's going to be splits, binary splits.
Yeah, but do you think that you can get a phylogenetic tree with fully radiation events, with a fully polytomic?
Yeah,
Phylogenetic tree?
fully polytomic, no, but with a lot of polytomies. That they radiate in very short period of time. But you cannot reconstruct each one of them, and then you have this polytomy.
Yeah. This is a couple of nodes.
And the last thing, to make I don't like this word of evolvability. Maybe capability of diversification.
Yeah. This is
Well,
a fancy word.
no. It's a matter. It's not.
Right.
And the thing, everything you got, if for make inference of diversification, I mean, if you have more birth of lineages or more extinction of lineages, first of all, and you are going to publish in any journal, biological journal, you need a full sampling of the extant species.
Yeah.
Otherwise, you don't know if it's extinction or because you didn't sample. That's all. I mean, all these things. And for that, I think you should try to take a good data set of a full sample to make any kind of these results, because otherwise, with the full sampling, even the full sampling is wrong, because you don't include all the fossil and all the past species. At least having all the ones.
And you know that I was looking for.
It's not many. It's not many. Very few.
That's very difficult.
Yeah.
But yes, we are looking for that. And the thing of evolvability term, I don't know.
No.
Evolvability concept, I think that this is very new in evolutionary biology, and I don't know if this is real or not. But it's a way to call it.
Again, congratulate you for this great job, and to the directors.
Okay, thank you very much. Now it's turn to Carlos Marian, please.
Thank you, Susan. Thanks for inviting me here today. It's really a pleasure. So I have 2 specific questions and 5 more general questions. The specific questions are quite, I think, related with the one questions about the data sets. So you showed a graph plotting the distribution for the X domain with F scaling, sorry. Yeah. So normally we assume the data has errors. We have some uncertainty to determine the position of the nodes and the topology. So did you try at some point to randomize or to take into account, instead of the mean values from the data, you could potentially assume a distribution around each terminal point, and then you feed the topology, not about it with just one tree per taxa, but a distribution of trees. So you don't have just one value, but a distribution of exponents per tree. Do you have a feeling, if you take into account just uncertainty from the data, assuming any general distribution, how this seems quite a normal distribution, right? It's a narrow variance. Do you think the variance is going to increase, or the shape of the distribution? Do you have a feeling?
Well, the distribution will be uniform distribution, I think.
You think it's still?
But for the real data, I don't understand. I don't understand.
Instead of just having one tree, you have a population of trees.
The population of trees.
Yes.
Yeah. And then measure the probability distribution.
Yes, for each population.
Population. No, then the same.
You
the same distribution, because we analyze this probability distribution also taking into account all the trees. Mostly. But the different set of trees and the exponent will show the same distribution. No, but I'm thinking, you mean measure fitting the exponent for the different, taking into account different trees, and then it's the same.
So instead of having them just one dot, you have
a
dot
no, they are the same. They are just the same.
The second specific question is I think specific, but could be potentially useful for any field. So there are currently several tools to get a livelihood among distributions, from broad scaling to power loss. What do you think, if you use maximum livelihoods comparing several distributions, do you think the power loss is going to be the best fit?
That's a tricky question. We don't know. As I said in the section of the models, there are different explanations, and which is the truth? We don't know. There are different models. As I have built a model that explains the transient behavior, I'm more confident in the transient explanation than in the other one, but I have no idea. Both of them, they have the same probability to justify the branching scaling. The problem is that those models, they don't have any biological mechanism that could explain the scaling exponent. So that's why I think more that based on this behavior and explanation, but we have to work more on that direction, because the fitting for this is what you said. We need more statistic, and the main important thing is that we have a big problem with the data, that we need bigger phylogenetic trees in order to characterize the large scaling level. That's an open question.
Okay. Great. I have 5 more general questions. I really have enjoyed your thesis. It's quite detailed. have now 5 general, I think, questions. Some of them are more kind of in the context of the philosophy. The first question is more is an old question. Is according with your results, micro and macro evolution are essentially the same. So you can predict potentially the whole tree of life just according with the same set of mechanisms. This is quite still challenging.
Yeah.
But then you develop a model, probability, or where you put explicitly differences among species.
Yeah.
At some point, you get a species that can speciate.
Yeah.
And so this is a species level property.
Yeah.
Assuming all individuals within that species, they have the same probability to speciate.
Yeah.
But then I started to think, okay, so why should jump to species level approaches to predict topology or any other property of this evolving, these diversification processes, when we still can keep neutrality at individual level? So if you assume individual equivalence, the classical Hubble model, then you have a speciation rate or the net diversification rate proportional to abundance of each species, without adding any explicit difference at the species level. And this, intuitively, this generates imbalanced trees. I don't know if the distribution of branch length is a thing. But what is your... So do you think neutral processes can explain the jumps from
The
micro to macro evolution? What do you think?
I think that our work is not enough to enter in the discussion if gradualism or saltationism. But for me, the main conclusion is that what we observe is a question of imbalance, of scales. There are mechanisms that are taking place at different scales. So maybe, for me, but I don't know if maybe it's what I learned during biology, that I think evolution as the unit of evolution for me is the individual in a population. So this is the classical gradualistic point of view. But I think also that you cannot discard macro mutations or different things or different rates of mutation between different species, or at different scales maybe you can have different population mechanisms that lead you to this general scaling at different levels of evolution. Why do we focus our model from the species level? Well, this is because intuitively most of the evolutionary models that we were using were based on the characterization of species diversification. This is more intuitive, and exactly, as biologists you can talk about populations, individuals, and make also in fact population genetic and everything, but from the point of view of a physicist, the idea is to get a model very simple with one parameter and nothing more, and that's the best thing that you can do. And that's why we propose this model, and a very simple model, and after using this mechanism, then we try to give some biological background, but the inspiration was not a biological inspiration, it was, you have two mechanisms that they give, explain the opposite behaviors, let's combine and let's see what happens. And we found that they give the same scaling, let's see which could be the biological interpretation. In fact, my first interpretation, I inspired this model contrasting allopathic and sympatric speciation processes. In the sense that I think that an allopathic speciation can give rise to two subpopulations with similar heterozygosity index, and then they have almost the same probability to speciate if you compare a population that is, or a species that is speciate with a sympatric speciation that they has a lower heterozygosity index, and then. But our idea was, since we found general scaling at gene level, at species level, our main goal was to see if this model could explain also not only macro evolution but also gene phylogenetic trees, which mechanism could relate our model with gene evolution. And that's why I thought that maybe we can consider a deleterious mutation as, or let's say, if you keep in time the deleterious mutation of a gene, and they are deleterious, they not appear in the population, but if you could reconstruct the phylogenetic tree, you can eliminate all these deleterious mutations, and this could be compared to a mass extinction event. But this is too general, and this is too theoretic, but this was the idea. But the main point is that this is a problem of scales. At a certain scale, the SNPs are relevant, and macro scale, the mutation of a whole gene is relevant. It depends on the scale. But one of my interests now is to go deeper in the biological background of this model and introducing population dynamics and everything in order to go in this journey.
Have you thought about key innovations as a potential mechanism?
About?
Key innovations.
Key innovations?
Key innovations as a mechanism of your model. It's like some set of traits in individuals that use, for example, better some set of resources.
But that's the point. For me, the idea now is, on the one hand, you have an organism, but starting from the model, we have the possibility that a given taxa is unable to speciate again. And what does it mean? That, in one hand, there is a possibility that there is a mass extinction event, but there is another possibility is that the selection pressure for this organism is very high, or it's so key innovations. We thought this in the sense to let's how can I explain this? You have a population and there is a selection pressure. So the population has a certain resources that can explode, and the problem is if there is some diversity in the environment that can lead to new diversification events, and this is something that we wanted to do, to combine diversity in the environment, selection pressure of competition of different predators and everything that can make that increase the selection pressure of the organism. But on the other hand, this is our motivation in order to characterize this robustness of a species. But on the other hand, we wanted to analyze this evolvability of a species in order to see if it's diversified or not. and in this sense, we want to consider key innovations in the sense that you get a new copy that with a mutation that is relevant for a new. For example, birds. No, the capability of flying is a key innovation in the diversification process. So that's something that we wanted to include. This is from the biological point of view, from the.
So I don't want to ask you all the questions. I think I would like, but maybe have one more, and it's a consequence of this discussion. There are two big frameworks that underline the idea of radiations and diversification. So one is the most accepted today is radiations as a consequence of a niche use, so niche adaptations. And this is essentially the idea of Darwin. But then, and the point is that this niche filling, for example, a boundary effect, and then you have a niche filling and a radiation. In the tree, normally you have the opposite pattern you have found. So you have the opposite of, I don't remember now the word, stemness. So you have the opposite. You have in the first part of the tree, you have and then you get a steady state because all the niches are filled, and then you get a steady state, and this is one extreme, and one explanation is essentially the niche filling. But the other extreme is sexual selection without niche adaptation. So you have frequency dependent selection, negative frequency dependent selection acting on individuals, and then you also have the same pattern. So you have a radiation and then saturation without the requirement of adaptations to niches. Strong discussions today, these two extremes. But this is the opposite pattern. And the recent paper by Venditti et al. Studying the distribution of branch length. So this is, you discussed this in the discussion section. You discussed about the problems with the data, because essentially you don't take into account extinct taxa. So what do you think? What do you think between the most accepted way most communities have radiated and the free topology and your findings?
Well, for me, the main point is that we have a big problem with the data resolution, because for me it's hard to imagine that this kingdom diversify that so few, taking into account comparing with all the Eukarya kingdom. So starting from this problem, I don't feel comfortable in order to make some biological conclusions in that sense, because I think that the main problem is that we don't have data to trust the biological background. I said in the conclusion, we can explain this as and for me, a good starting point is considered the radiation point of view, and for me it's one of the first that I was thinking in order to explain the distributions, since the most diversified groups are corresponding to these radiation processes. No? For example, plants and arthropods, birds, and those were the biggest groups not in this, because this is very, very small, but in the catalog of life, this huge one. The largest groups of organisms corresponded to those groups of organisms that undergone this kind of radiation. But I would say that taking into account that the taxonomy in microbiology is so young, so the Berg's systematic manual was published in 1923, or something like that. Yeah. The taxonomy in microbiology is very young, and I think that this tree is reconstructed in that way because it steps, the leaves are the families, so you are excluding a lot of species there. You have the problem in the classification way of microorganisms, and also the problem in the sampling. So I think that we have there is a lot of work for the organisms, for the biologists in the characterization of discovering new species at microscopic level. So then, I don't know about this theoretical thought. Yeah, intuitively I thought also about radiations and everything, but I think there is a problem without the excluding of mass extinction events and all the problem with the data. But also with the branch length, this for me is crucial also the exclusion of the mass extinction events that lead you to a stimulus pattern due to the fact that the older the tag size, the more probable that a species, that these tags are extinct. Yeah, that's open questions.
Yeah, I'm also essentially asking this question because potentially it could be great if you increase resolution and the pattern remains the same, then we don't have to invoke niche adaptation or sexual selection. Maybe we can have potentially a new set of mechanisms as an explanation.
Yeah.
I mean, I don't want you to tell me this mechanism. I believe. I mean, yeah, but I'm done. Thank you and congratulations. Yeah, to the team. It's a great work.
Okay, thank you. I'd like to invite Dr. Catherine Marchal to put your questions.
First, congratulations. I really like the work. I think you really convinced me now of the use of complex network theory to analyze biological problems and physics in general. So I think my first question also relates a little bit to this biological interpretation of these micro and macro evolution events that you have in your model. At micro evolution, at species level, I can somehow understand. But what does your model mean at gene level? I mean, that there is a stop in evolution of a gene.
Yeah, that there.
What does it mean biologically? Because the gene is still present in all the other species.
Let's say this is a particle. From the physicist's point of view, this is a particle. You can call it gene or species. It's it. So, if you consider a mass extinction event, this is erased. It doesn't exist anymore.
So you really mean that the gene is erased, not that it's evolution, that it stops evolving?
No. Now I'm talking about macroevolution, and then I extrapolate to...
Yeah, the macro,
I know, but it's almost the same. At microevolution, then you have this group of organisms and you erase as a mass extinction event. And then you keep only this organism. But at a certain moment, all the organisms were existing, and then mass extinction event, all erased. From at the gene level, what I think is that you have a gene, and this gene suffers some mutation events. But those individuals with those mutations, they don't live.
Because the species were raised.
No, at the individual level. So this is a deleterious mutation.
Okay.
So for me, these were like deleterious mutations that, since they act at the zygotic level, you are not able to see.
Yeah, but that you can never see in the trees that you analyzed, right?
Right.
Okay.
Yeah.
So how can you explain in the trees that you analyzed?
Because since you don't see this in the trees, what you see is that this gene is unable to give rise to new genes, and so that, let's say, the selective pressure is very high in this gene, so it's what we call is very robust, while at this level the different genes are able to diversify, but this gene has a high selection pressure.
Yeah, okay. Yeah. Yeah. And then it's somehow by now generally accepted that genome-wide duplication events would give rise to new species and new, they generate new genetic load, and that can give rise to new species. Can you somehow take that into your model as a kind of explanation, or can your model handle these events as it is now? Like at a certain moment your whole genome duplicates. So we would expect suddenly a lot of new branches somehow, but then most of those will be mutations that fastly decay exponentially almost because.
Well, the problem is that the duplication of the whole genome is a punctual mutation event. Yeah. So in our trees, this is only one node. So it doesn't explain the average pattern, and this would be related with a certain subtree of the phylogenetic tree. And well, since
why would it only be one node? Why would it only be one node in the gene tree?
No, one node, no, but a set of nodes, but a certain subtree. And I probably don't even see it anymore. But it could still be a generating mechanism of. No, for sure. But the second part that I wanted to say is that I think that mutations, what we call the capability to diversify, this is at different levels, and the mutation that is originated from the duplication of the whole genome is like a macro mutation. This should be represented at a macro evolutionary level, at least. So, for our work, how can I explain this? We analyze phylogenetic trees at population level and phylogenetic trees as species level. It doesn't mean that both phylogenetic tree datasets are reconstructed with the same biological mechanisms. On the one hand, maybe there are small mutations that they are reflected at the population level, but at the macroevolutionary level, the mutations are large-scale mutations.
Yeah, but that might just be a kind of mechanism that works at the micro level that could explain the macro level.
Yeah. But the effect, the biological effect over the organism is reflected at a macroevolutionary level.
Yeah.
So after a certain time, the duplication of a genome is not trivial for an organism. So this would give rise to a new species.
Yeah.
And this,
maybe
is
and another question related to the way you model the mass distinction event. So you delete all the nodes below.
Yeah.
But isn't mass distinction just meant to erase almost all species except a few that then go through a bottleneck and go into speciation? Because now you erase them all, so I thought it was like a bottleneck effect
and then the living organisms are occupying the...
Because you reduce the genetic load and then you have...
Well, the definition of mass extinction, yeah, is the elimination of...
but then nothing would exist anymore.
Well, it depends. You can see, yeah, in a dynamical process, you can see in that way that, and this is something that we want to do now in order to analyze the branch length distribution in our model. But this is future model. But the point at this level is that you can say, I have a mass extinction event and then a bottleneck, the bottleneck effect, and then the increasing in the diversification rate of the other groups of organisms to occupy the different. But you can analyze this problem in the other way, that you have the whole tree with the fully reconstructed tree, including the extinction process, and then you erase the taxa that has gone extinct. And this is the approach that we...
Yeah, but then when you model, you don't take into account that there is like maybe an increased branching of the ones that are left. So you do it after the generation of the tree. So...
Yeah, well, classically, but this is a fallacia dominem, that usually the people model the mass extinction event in that way. You reconstruct the tree and then you erase.
That's fine.
And this is, well, for me it's almost the same. So, okay, because now this is the classical example of the dinosaurs. So you erase all the group, the subtree of dinosaurs. This is a mass extinction. But
yeah, but then the mammal is evolved much faster than previously. So it had an effect on the others. So, and if you just take it out of the tree, if the tree is not made on real data, if it's made on real data...
Yeah, but it doesn't mean that it will be represented in the branching pattern. Maybe this is represented in the branch length.
Yeah, in the length.
But not in the branching.
Yeah. Okay. That's it.
Okay, thank you very much. And now, Dr. Montoya.
Okay. Well, control limitation, I think it's a really nice piece of work and a really nice collaboration between biology and physics. And I'd like to congratulate you because of several reasons. Well, the first one is, I think it's a very original and creative thesis, and I think this is basically one of the reasons why we do science, because it's a creative process and you have reflected it very well. The second one is very ambitious thesis, okay? And probably it's more ambitious at where you can get with the data that you have, but the questions are really, really ambitious, and this is a very good thing for a thesis. And the questions are very clearly stated, which is a very good thing. Then the methodology that you use, of course, I'm a fan of it because I like complex network approaches, but I think it's very, it's original and it's very, it goes with the questions you're asking, so great for that. And it's very well written as well. And then I think it's very relevant, the questions you're asking. And of course I can be biased because of that, but I ask people working in phylogeny at my research institute, which are no fans of complex theory at all. And they say, well, probably we'll contact him to analyze the phylogeny that we have. So, I mean, it's relevant even for people not familiar with these approaches, which is good. Also, I mean, the quality of the thesis is already contrasted. I mean, you have 2 papers published, and one of them in PLOS biology, which is good. And I think it's brilliant also the knowledge that you show from the area. I mean, I love to see these tables with the ontogeny of the ideas, and probably I don't agree with some of the selection of the things that you put there. For instance, why in the last 40 years the last good contribution to evolution is in 1972, I think. So it's been 40 years we haven't contributed any relevant point. So this doesn't show a lot of good things about current scientists. But it's great. And another thing, in a more personal point of view, I mean, you show a lot of honesty in the way you present things. For instance, the appendix B that you present, and the appendix B saying that they criticize your paper in cause biology. I mean, you didn't need to present it, okay? But you've done it, so this is very good quality of a scientist. I congratulate you for that. And I really like the structure, though at the beginning I was a bit lost with all the appendixes and jumping from one place to another. So it's probably the only criticism I make to this structure. Probably. I mean, some of the appendix you could have included them in the chapters. I mean, it's a minor point. It's nothing. And well, now some questions. I have many. I'm just going to ask you 3 of them. In general, I've missed some statistical robustness in some of your conclusions. I mean, basically there are no statistics in the thesis. And I mean, at some point you don't need it because the data are very clear and the models are very clear. But when you use allometric scaling and you read papers about allometric scaling, every time you fit a line to a group of dots, at least you need to provide a confidence intervals, for instance, to see. And this can help you a lot to see whether it fits a particular model or not.
Yeah.
If you have the same exponent, you have to say whether it's the same or it's different, and you have statistical tools to do it. I mean, allometric scaling studies, most people do. And for instance, when you present in chapter 6, the different exponents for the rank base, rank 3, etc., and you say, well, these are closer, these are more. It's a lot of. I mean, people can criticize it's a lot of blah blah blah, and you have statistical power to say how different they are, so can improve the robustness of the results. Why you didn't use this thing?
Well, this was more a qualitative analysis, so such a quantitative to compare. For us was evident the big group of differences, and it would be nice here to include some statistical, but
when you compare to slopes, I mean, you have ways of saying how different they are.
But we tried some, but I think that there is also a problem with using it.
Well, in general, physicists doesn't like statistics.
That's what I wanted to say. But I think that.
But
I don't want to.
But when you use real data.
But there is the jokes between physicists, mathematicians, statisticians.
So it wasn't your problem. Okay. Then about the data. I mean, we can discuss about data all day. I mean, it happens with every meta-analysis that you do. Data are incomplete, and sometimes you mix wrong data with proper data. I mean, people that reanalyze the conclusion of some meta-analysis that some people have done, there's like 60% of the conclusions that were wrong because they didn't select properly well the data. But in particular, the data, I'd like to ask you a couple of things. The first one is that why I'm not saying during your thesis, but probably later, why don't you select the best quality data that you have and perform the same analysis? In that way, for instance, using instead of the 3 methods of reconstruction that you use, choose the best one, which again, most people, maximum likelihood, just use this type. Now that we have computer power, construct the data with that and reanalyze, so people won't criticize the kind of data that you use. So select the best one and use the best reconstruction method. Do you think it will hold the results that you found?
Let's see.
When you select the good subset of data and you use the proper, the best reconstruction method.
This will reproduce the same result?
Yeah.
This was a general depiction of all the data sets, but we analyze them separately and taking into account, comparing also, trying to analyze the full phylogenetic trees of full genomes, and they are taking into account all the problem with the statistical of the data set, and we observe always the same analysis, the same results, but the problem is that the number of data are so small, so few that we cannot show it as a relevant result, but the result, the scheme was the same, not the difference. And the best one is the maximum likelihood method?
Well, I mean, we can discuss about it as well, but when,
I don't know which is the best one.
Yeah, well, okay, we don't go. Because I'm saying that because, I mean, some of the, I mean, your model fit and all discussion about the transient behavior due to that some of the data are really small phylogenes. So when you have big ones, I mean, you won't have that problem. So I'm saying that because some of the other patterns that you find could be affected by that or not. So with this argument, you only made it for the you only make the argument in that part of the thesis. With included large data set, we eliminate your transient behavior, blah blah.
Yeah. Well, the problem is that this problem with the size, not with the statistic of the reconstruction method, but with the statistic of the set that we have, that we larger sizes.
Okay, well.
No, I don't know. Maybe I don't
No,
understand
it's
the question.
it's fine. The other thing that in the figure 4.5, you put a figure here where you have it's 4.5. Yeah, I mean this example. Yeah, here you said that these dispersions are mainly due to the risk. I mean, basically that the phylogeny is not well resolved, so you have all these roots to the base. Mm-hmm. But I mean, I was thinking, and lately I'm very involved in this kind of works on tight coevolution between species. Okay, so you can think. I mean, you talk about parasites. But I'm thinking about endosymbionts. When we talk about endosymbionts, I mean, in sponge and corals, you can find between 500 and 1,000 different bacteria that are symbiotic within them. And you can see there is, I mean, at some point, they establish there, they go evolve, they reduce the genome. So if you make a proper gene phylogeny there, you will see that they are attached very, very, very deep into the roots. So they establish and they co-evolve with the host and they didn't diversify anymore.
Yeah.
So I think it can be a pretty common pattern in this system where tight coevolution is very important. So do you think that will be the case?
But once again, the main goal of this thesis was to characterize global properties of the phylogenetic trees. I'm sure that there are a lot of biological mechanisms in order to explain the different oscillations that you can find. But again, yeah, this could be an example, or for example, this and this could be also some prints of some evolutionary processes, different, but these are local behaviors, but what we wanted to analyze is only the global scaling, because in order to analyze these local behaviors we need more time than this is. But I agree with you that I'm sure that this, in fact, my desire at the beginning as a biologist was in order to identify easily differences from the biological point of view. And what I thought was this universality and always universality. But I'm still with the idea that we can identify some, but the important thing is that we found a universal scaling, and these low differences and this effect of biological mechanisms are reflected, but these are only a set of points in the tree, only a set, that they don't affect to the average scaling of the tree.
I can see that. I was just making this argument because, I mean, your argument was that you find this because you have poor, I mean, this is not well sampled. Sample. I mean, you can get a lot of these deviations that are not due to lack of data.
I didn't know that about evolution. Thank you.
And well, we're not going to talk about the species concept in some of the work they present, like bacteria, archaea, etc. But it will. I mean, it's the problem with these global approaches. I mean, when you go into there, it's a bit problematic.
Yeah, that's why I used the public paper. So I'm not saying that that's a species in bacteria.
And well, that's not for question. The only thing is, I like asking is, which part of the PhD is more yours? You feel it more? I mean, a PhD is a co-evolution between you and your supervisors. Which part of it you find is more personal? I mean, which kind of finding you would say, well,
Should we leave the room? Let's see. All this work started with this one, and during the first year I was by hand. As a biologist, I was not able to write a code, so I was nothing about mathematicians, nothing. I was doing everything by hand, and well, in this work was let's say the hardest, but I was very present in the compilation of the data and everything. This and the compilation of gene taxonomies, and then after the time tree, this was with the collaboration with let's say with people from here, and it was more in the I would say in the modeling was more my point of view, because the main part for me is the modeling, because this gave a sense to the thesis, because till then it was only describing data and nothing more, and I wanted to explain, give some biological explanation, and that's really hard. For me, well, I take part from the biological and from the obtention of the data, from the analysis, but all the part of the mathematical point of view, this was all my supervisors that were by osmosis or active transport leaving me, but as from a personal point of view, well, I think that the most personal was the modeling.
That's all.
Okay, so now it's my turn, and So again, as other members of the jury have already said, my congratulations for the work. I see evolution while I was reading, and I was very happy to see that some of the questions that came to my mind during the reading process were later answered. So you had the same questions, and you were really fighting to get some answers so as honest and as close as possible. I also want to congratulate you, especially apart from your supervisors, because you are a biologist and you were brave enough to join a team of physicists and a physics institute, although your degree will be in biology, but still, I mean, you've been working with them. And I know from my experience that this is really the only way that we have to advance in the interdisciplinary research of these fields, right? So, I mean, this is a nice effort, and I really encourage other people to do it, because otherwise we will always be in our particular communities without talking to each other. That's essential. And finally, also my congratulations, because you have convinced me. So you know that at the beginning I had some, I had some criticism about the patterns you were seeing and the interpretation of those patterns. And you have managed to find quantities that demonstrate that you have a different process, a non-trivial process, constructing the phylogenetic patterns that we see in evolution. So I think this is a tour de force with your data also, and in the interplay, in the developing of measures that capture the processes that you see. So in this sense, I also believe that probably the mechanism behind is simple and universal enough to generate these patterns, and this is still, I understand, an open question in the research. So this was the beginning. And now I want to go to some specific questions on your work. So at the end, I don't know if we, in the point that we have agreed about this, all the work you have developed, do we agree on the crossover to the random behavior for large enough trees? I mean, you see it in the model. I think still that this is something that has to occur, because mutations erase the memory of whatever properties, species, genes, or any other taxon have. But there are two questions. Your personal opinion about this? Well, there are three actually. What's the crossover that you could see from the model, and whether the current data can give you a hint of where this crossover lies or not? Personal opinion.
Yeah, but repeat the question.
It's about
because I have to. There are some small details that. Yeah.
It's about, or maybe it's about the crossover to the random behavior.
Yeah.
So I mean, you have convinced me that there are non-trivial correlations up to a certain time scale.
Yeah.
And or tree size, if you prefer.
Yeah.
And now the question is whether you think that there is a crossover to random behavior. First question.
I think so, because, well, taking into account that for our model, we found this random behavior at large sizes for a big space of the parameter. So I think that this behavior is so powerful, the random scaling is so powerful that at a certain size you will fix to the random scaling. And then I think that these correlation processes are going to be a small.
And how far do you think that we are from having data enough to see whether this occurs in the real process?
Real data, to get real data. Well, with all the Crime-Penter and all these big projects, with full genomes analysis and everything, and all the technology that we have, I think that we can get soon large data deep trees. The other question is the quality of the statistics and the problem with the extinct data. Because that's the problem. The easier you get the data, the more problem that you will have with the extinct data, because with microorganisms it can be very easy to get a very large tree, but you don't have there. You don't know what's an extinction there and everything. So, answering your question about the data, if we are able to get large trees and so on, I think yes. But
so you think 1 million, the last data you have, is not enough, or is not reliable enough to show this change of behavior? The catalog of life?
Yeah. In order to what? Do you mean?
Yeah, so here you still don't see any trend to change. Yeah.
For the catalog? No.
Because you have very little also statistical powers.
No. And this is a taxonomic tree. This is not biology.
Well, okay. I'm not so strongly worried about the data, because when you see the universal pattern everywhere, this means that they have something in common. It can only mean that what they have in common is not as relevant as one would like.
Yeah, I was joking, but in order to analyze the depth scaling of the phylogenetic tree, the constraint of the taxonomic criteria is so hard that you cannot compare the taxonomic tree with the phylogenetic tree. So then I don't feel comfortable if comparing these. No.
Okay.
So this is different stuff.
So then it's for the future still. Yeah. Okay. So my next question is somehow there was already some discussion on that. When you have introduced your model to try to mimic what you observe, you have considered internal variables for your species, genes, whatever, for the nodes and the branches of your trees, right? This is P1 minus P. This is something that you can inherit and is passed to the next branches, so to say, right? So this is a point of view that I also explored many years ago, and this is the species from inside producing the patterns that we see. But another point of view, of course, is that the environment is what is limiting your branching and so on, and actually niche filling. And if you look at the whole history of metazoa, you see that towards the present you have more diversification of the branches. And I think this is a robust pattern, irrespectively of the not knowing enough about extinct families, for instance, right? So, have you any idea of how these constraints from the outside, environmental constraints, could be included in a model that would be then reflecting also this kind of behavior? And this is also related to your last part, to the branches becoming shorter towards present. So it's a change of viewpoint, and then you don't have to rely on internal processes, but either on coevolution or on niche availability on the model.
Well, what I tried to modeling the branch length distribution. I tried to model it using our model, and what we observed is that for the random case, you don't see any correlation, and in order to get some correlation, we obtain some correlation increasing the refractory period between different
but it's
diversification events.
Somehow still from the inside, or I don't know, how do you interpret this?
Well, but a species is not like Ortega-Gasset is the species and the environment. No?
Right, that's the point. Yeah.
And I would say that I'm not saying, I think in any part of the thesis, that this is a property exclusively from the species.
Mm-hmm.
I consider that this is an interrelationship between species, the species and the environment. But obviously, in the model I say the species is able to evolve or not, but the capability to evolve or say the incapability to evolve, I don't know if this is something related with some biological property of the. In any case, I would say it depends on the relationship of the species with the environment.
It's just a suggestion.
So
it would be nice to think of a model.
One of the points that we want to do with the model is to analyze the correlation, changing the environment and considering diversity in the environment and how the interplay with the fitness and selection, but including population dynamics.
Okay. So then some last comments. I'll go to one of your appendices. Nobody has touched upon the appendices. And I'm also I'm very much interested also in linguistic evolution. And I think that there are so strong parallelisms in some quantities, so not only qualitative but quantitative. We know from the literature, from published results, it's not something new, that it makes me wonder whether this could be an aid to understand biological patterns. Because what is not in the linguistic evolution, then if the pattern is the same, cannot be in the biological evolution. Sometimes fine details that you ascribe to one of the systems, if you see the same topological pattern, then you know this cannot be the force shaping it. Right. So I think it would be nice to go further into this direction. And I have, however, some specific comments. Did you want to say something about the?
Well, and in fact, this is an appendix because it's an embryo of the work, and our motivation is to follow the line of different research groups that they are characterizing diversity, language diversity, and species biodiversity, and all this related stuff.
There is a work that you may not know of. It was done by Damian Zanetti, and he was taking this degree distribution, so the polytomies of languages at different taxonomic levels of languages, right? It's not there.
No,
so, and it's very interesting because you see that when you go down in the taxonomy, the degree distribution changes slope. It's a power law that with a slope that depends on the level of the taxonomy.
Yeah.
And I don't have any clue about this, but I wonder whether there could be a relationship between faster speciation events or slower, right? When you move across these levels, and whether there could be some parallelism again with what you observe in biology. So this is just something to think about, and if you
So the polytomies they're more present at higher level in the taxonomic.
Well, you have to look at the distributions because he was giving only the degree distribution, and it changes slope with the taxonomic level.
But
it is also something that you can measure easily in biology and see whether there is a correspondence as well.
No, but that's
So it's
but I'm thinking about the political interpretation of this. No, political linguistic.
Anthropological, you mean?
Anthropological,
The linguists classify. Okay.
But
Okay, so
that's
everything. many issues to talk about, and I think that we have had a very long discussion precisely because you have touched upon things that are of interest, different disciplines, which are not closed, and that's very nice. You're opening some fields. So, congratulations again. Okay, so now is any of the doctors in the audience, among the audience, wishes to make a question? No? Okay, then we thank you. This finishes the public session. We will now discuss.